right, well, I think we're going to start. So we are really happy today to have Dr. Lachke from University of Delaware to talk to us about his research. Before I introduce our speaker, let me go over some housekeeping issues. Please make sure that you sign in. The signing sheet is on the table just in the back here. But for next time, please RSVP so that we can better prepare for the next session. Online participants, please keep the mics muted unless asking a question and keep video turned off. The next speaker will be for our tech talk on June 2nd, and that is Paul Nitter, who will speak on constraint randomization. And then for the innovative discovery series, we'll have optimizing values through appropriate use of invasive cardiovascular services by Dr. Garratt on June 3rd. And then on June 17th, we'll have Lisa Cooper from Johns Hopkins coming to talk to us about reducing hypertension disparities. You can see the full schedule on the ACCEL website, and all the previous presentations are available on our website, DE-CTR.org. So we are really thrilled today to have Dr. Lachke. Dr. Lachke is Assistant Professor in Biomedical Sciences at University of Delaware and also a Pew Scholar in Biomedical Sciences. He obtained his Ph.D. in Biology from University of Iowa and his Master of Sciences in Microbiology from University of Pune in India. Prior to becoming Assistant Professor at the University of Delaware, Dr. Lachke was Research Fellow in the Department of Medicine at Brigham and Women's, and also Instructor of Medicine at Harvard Medical School. He has had multiple awards and recognition over the years, mostly related to eye research, and he is and has been the PI on several R01s. One R01 which recently ended called post-transcriptional control of gene expression in lens development. And he also is currently just received a new R01 from the National Eye Institute for a project entitled Post-Transcriptional Control of Gene Expressions, also in the Institute. And he's also the PI on a very large R03. He recently also obtained a prestigious 4-year grant from the Pew Charitable Trust in 2012 and has multiple grants from foundations for eye research, such as the Knights Templar Eye Foundation, the Fight for Sight Foundation, and others. He's been the mentor and supervisor for numerous graduate students, postdoctoral fellows, as well as undergrads, and has a very long list of publications. So it's really an honor to have Dr. Lachke here with us. Thank you.
Can you hear me? All right, fantastic. Thank you very much for such a nice introduction. I really, really appreciate it. So I'm quite excited to be here and share with you, research that is going on in my lab, but also try to sort of put it in the context of how it can be really translated to the clinic. So first of all, I just want to make sure, where do I see really? Do I just see the audience here? Is that good? Should I look at a camera or what? This is fine, right? All right. So today I'm going to talk about several birth defects that we have used some fundamental biological approaches to gain insight into, and most predominantly the talk will be centered over congenital cataracts and some of the other eye diseases such as Peter's anomaly or anophthalmia or microphthalmia, and a little bit about how we can make a splash into cone rod dystrophy and retinitis pigmentosa and the Bardet-Biedl syndrome. And eventually I'll wrap up with showing that the approach that we are using is relatively straightforward, that it can be translated to other organ and tissue systems. And the one that I will focus on is going to be the orofacial clefting. Okay. And so really, what is the challenge that we about doing human genetics right now, even with the latest technologies and such? It's really like finding a needle in the haystack when you're talking about having that one mutation or identifying that one mutation in a patient. Okay. And the reason I say that simply is because there are 3 billion with a B base pairs in our genome, right? And of course, 37 trillion cells in our body. That's astronomical amount of genetic material. But of course, we can sequence it, right? And you will see that our sequencing strategies have been so well in the last few years that we have been able to now use next-gen sequencing to really under $5,000 sequence a human genome. But of course, the most important part is not that we can do it. What do we do with that kind of data, right? And imagine if we can have this sort of data from every patient. It really comes down to enormous amounts of data, quantities of data that somebody has to go through and try to come up with that mutation or meaningful sort of data. So the straightforward genetic challenge that we have right now is that how do we prioritize the candidate genes from whatever variations that we see in a specific patient with a certain anomaly, okay? And essentially that is, adding causality to variations, right? Which variation is just a normal variation that we just happen to find right now, and which one is really associated with the pathology, okay? So essentially it is that you can sequence genome, you'll get, lines and lines of that, but how do you go from that to finding that's one base pair nucleotide change that leads to a congenital cataract, such as in this 2-year-old patient right here. So before I move on to my sort of part of the talk, I want to just tell you guys that in this particular month's issue of Human Genetics, the journal Human Genetics, there's a fantastic set of reviews on exome sequencing. So anybody who wants to know about exome sequencing and how that can be utilized for birth defects as well as cancer and such, and what are the challenges and what can be done right now, I think I will encourage anybody to go and read this entire issue, in fact. So now let's get back to the topic that I'm interested in, and I'll try to show you how we can use these approaches in combination with animal models to try to really influence gene discovery in humans. Okay, so we work on the eye and the orofacial morphogenesis, and so let me introduce the eye first. And very simplistically, it's a camera-type instrument really in our body, and it's got a cornea and a lens in the middle and a retina at the back. And the cornea and the lens are really the organs of refraction, which will reflect the light into the eye for maximum refraction onto the retina. Okay, and this can be really best illustrated instead of a physics diagram and such by just showing you what a mouse eye looks like when we shine laser beams onto this. And you can see on this top image that all these laser beams can converge onto a single point. And of course, even a slight aberration into this morphology of the lens, which is in the bottom image of a mouse mutant for a specific gene, you can see that sharpness is already lost. And of course, the lens depends upon its being transparent, completely transparent as a tissue for this function of focusing light onto the retina, right? And when it ceases to be transparent, which turns into a cataract, that's when people basically, unfortunately, have a visual impairment and go blind. And yes, cataract predominantly is an age-associated disease. That means by some numbers right now, this age of 80 is even getting lower by the latest numbers. But NEI right now says that if you are 80 and older, there is a 50% chance that, or half the population 80 and older has gone and undergone a cataract surgery or will undergo cataract surgery. But of course, cataracts are also found in congenital cases. Okay, when babies get cataract, newborns get cataract, and that's because of a 50% chance that there is a genetic basis for that. Okay, and of course we know that there are metabolic conditions that can give rise to cataracts as well. So of course currently the therapeutic implication for cataracts is that you take out the bad lens and you put an intraocular lens, an artificial lens, and that's it. And it's a very nice surgery. In fact, people have really used it amazingly to give sight for millions of people around the world. There are some really heroic sort of ophthalmologists and cataract surgeons out there who are doing some amazing work on this all over the globe, for that matter. The surgery at some of the experts' hands takes as small as little as 4 minutes. Okay, in 4 minutes you can restore the vision to some person, or it can be a little longer depending upon the case. Okay, but the point here is that you are replacing the lens with an artificial lens, and many a times this surgery is performed on patients which are already quite old. You don't want to undergo surgery when you're old. And also the other thing is that the lens always talks with the cornea cornea and the retina as a biological entity. And when you replace it with a plastic intraocular lens, that ceases to happen. And eventually, there are other complications that are that can turn up. But for now, cataract is has a very good remedy. Okay, so why do we study it, right? So we study it because we try to see whether we can do better, and hopefully by the end of this talk, I'll try to show you some very, very latest developments into cataract research, as latest as just a few months ago, which are published. Some of it is published from my lab, and some of it has been published by other people across the world, really. And we'll try to see whether we have alternative sort of therapeutic implication. So. Besides that, of course, the opportunity to study this in patients is to obviously know more about ourselves, right? Currently, we use animal models to really understand, like mouse, for example, to understand really how our organs develop, right? But the only opportunity we have as humans to learn more about ourselves is when, unfortunately, when there is a defect in a person and we try to find out the genetic basis of it. That is really the only way we can find out about whether this particular pathway or a gene is important in humans. So it's really informing ourselves about more about ourselves, that we can use this technology and try to then, hopefully influence therapy as well. And hopefully by the end of this talk, you will tend to agree with this. So let's start just a little bit of an introduction to the eye and lens development. This really started over 115 years ago when Hans Spielmann, who is a famous German biologist who went on to get Nobel Prize, he basically showed for the first time that the lens and the retina, as they are being formed in the embryo, are already talking with each other. And that means that they influence each other's development. And this is really just a series of set of developmental stages for the mouse, but essentially it's just very similarly shown to happen in humans. Okay, so essentially it's the optic vesicle, which is the outpouching of the brain, which interacts with the overlying ectoderm, which is the surface ectoderm, which is yet to form the skin. But that particular ectoderm is signaled by the optic vesicle, shown here in this orange globe here, to now form what is called as a thickened lens placode. And that lens placode starts forming a pit, and you can see it completely in pouches, and then whatever surface ectoderm can reform the surface will turn into the future cornea. That globe that has been formed here in the middle is going to form the lens, and of course the optic vesicle then goes on to form the retina. Now, interestingly, the lens is an organ that does not stop growing throughout our lives. Okay, and it's got a stem cell-like population in the front end of it. Okay, and that will differentiate into what is called as fiber cells in the posterior part of it, and those are shown in green here. So these are the embryonic stages. That's why they're shown in green. But you can see that in the last figure right here, which is that right there, you can see that those are shown in blue, and the reason they are blue is because this process of secondary fiber cells being differentiated. So the stem-like cells on the front end of the lens exiting the cell cycle and differentiating into what are called as secondary fiber cells, that occurs throughout life. So what we learn about development or developmental genetics can be applied later, and maybe we could use some of that knowledge to also think about why we get age-related cataracts, okay, or some of the factors that influence age-related cataract. So another bit on the lens, again, a different section, but this time from a developmental biology standpoint. So the lens has the potential to teach us some of the fundamental principles that are used in developing systems in other parts of our body as well. And I'll tell you why I say that. Is because in the front end, as I said, this epithelial tissue, which is the stem-like cells shown in blue here, and they transition into these fiber cells, which are shown in green. And these fiber cells start elongating. And look at some of the cell biological really challenges that this particular fiber cell has. It has to elongate. It has to build an arsenal of crystalline proteins, these specialized proteins at very high concentration levels will not come out of solution. Okay, and essentially that's why we are seeing each other right now. And of course it does that while it's degrading its organelles and its nuclei. And that's important because this is a cell which is essentially in the process of killing itself in some sense, but it does not really go through that completely. Okay, it builds up all the arsenal that is necessary for us to see through this really beautiful structure called the transparent lens. From a developmental standpoint, from a developmental biology standpoint, I can expect something like this, that some genes, some proteins will be expressed in the cell layer, the epithelial cell layer, which is a stem cell-like cell layer, like FOXC3, for example, is a protein that does that. And there will be some proteins which are expressed in the fiber cell layer. This is a very straightforward expression pattern that I think a lot of people can explain by transcriptional regulation of a certain gene, a promoter or an enhancer driving that gene or such. But then there are some very interesting expression patterns that we have noted, like this very important cell cycle kinase inhibitor called P27. This is very fundamental to cancer research as well. This is a molecule that will stop the cells from dividing. It will go and bind to a cell cycle kinase, inhibit it, and it will take the cells from an active cell cycle and start its pathway towards differentiation. Okay, so this protein, you can see, is very specifically expressed in this zone, particular zone in the lens, and then it comes down again. Okay, so this dynamic expression pattern in cells which are going to degrade their nuclei soon is very interesting. Okay, and of course, if we find out what are the regulatory principles behind such regulation, they can be used in other parts of our body as well, or to study other tissues and such. And of course, beyond those two expression patterns that I just showed you, there is this other expression pattern for an important heat shock protein called HSPB1, and you can see it's very specifically translated in some sense into the protein in these late differentiating fiber cells. Okay, and hopefully I'll show you that how today we could make really tools that could influence human genetics. That means we could find completely new genes, unprecedented, that we would have never looked for in the eye, and how that discovery led to really understanding of some of these patterns that occur in the lens. And in turn, as I said, how those principles that we have learned right now, how those can influence into other tissues and body parts. So this is very exciting time to be a biologist, I would say. So anyway, so really essentially, how does the lens control all these specific expression patterns? That's a biologist question, right? But I started with that question and then tried to really dive into this new gene discovery. Okay, so really I asked the questions: how could I design an approach to identify new genes in lens development? Could I try to see whether these genes that we would identify are related to cataracts or linked to cataracts or such? So how does one go about doing that? So I started reading the literature on the lens, really, and I started compiling all the points data points, that is, functional data points that there are into really systematically into recording them into Excel spreadsheets. Eventually, basically, we started writing algorithms whereby we could start seeing signals, in fact, very similar to what an engineer would draw, an electrical engineer would draw an electric circuitry diagram. And you can see that some of these signals from the retina, which is this tissue out here, which is the optic vesicle, is going to signal to the prospective lens right here, out there. And then eventually you'll see that just as the lens goes from these two different developmental stages, this circuitry just completely explodes in some sense, right? So we understand lens development, at least when I started doing this a few years ago, to this detail, at least these first few stages, we understand them quite well. But then I noted that the reason we came up with these kind of genes was because we depended on Drosophila geneticists who had identified, say, a certain gene in the fly first, and then we went and found the homologue in humans or in mouse and then tried to study it. Or we had depended on Jackson Labs finding a cataract mouse mutant, and then we tried to map the gene and find the gene and such. Or we depended, of course, on human patients in which there were cases of inherited cataracts. We could again map the particular mutation and then sequence it, and we could find those genes. So it was in largely indirect ways, I would like to stress, of coming to the most important players that can be important for lens development, and then trying to build this circuitry diagram, right? So then that's why we asked the question, could we do better than this? Could we try to use the technology of the time to try to come up with approaches? And so what do I really mean by this new strategy, integrated approach? It's really conceptually simple. First, why do we need it? To effectively identify new cataract-associated genes to gain insight into their molecular function. That's pretty straightforward. So what do we already know? Just to recap, we know that there is a wealth of functional data out there. There are over 100 original research papers really defining molecular interactions between different molecules in the lens. And we also know that there is a lot of high-throughput expression analysis that is being done. There are over 70 data sets out there right now that you can just really plug into the NCB PBI and start downloading on your computer. They're freely available to everybody, right? So these are data sets, they're expression data sets in the mouse lens, and some of them are in the human lens as well. But there is a lot of data. That's really the point of these slides. Okay, so essentially our hypothesis is very straightforward. Can we use this enormous amount of data, the expression data, the molecular sort of biology data, can we use it to really build a tool that can start predicting genes that we can start testing in animal models or go back to human patients and see whether really these genes are mutated in human cases, and that's why they got cataracts, really. It's very straightforward as a principle, right? So the first things first, right, we constructed a transgenic mouse which has a specific piece of DNA which has almost remained intact from fish to humans, and the reason it's remained intact is because it's very important to drive a critical gene called PAC6. If you knock out PAC6 in mouse, or if you have a homozygous mutation in PAC6 in humans, there is no eye development. Okay, so that's how profound this particular gene is. In fact, they're even more profound. If you take a mouse PAC6 gene and if you misexpress it in a fly, which is a Drosophila, you'll get an entire fly compound eye where it's not supposed to make it, like in the place of the antenna or such. So that's called as a homeotic mutation. Just as an aside, this is a fundamental gene, right? So the non-coding region of the DNA that controls the expression of this gene is also fundamentally conserved from fish to humans. So we use that piece and we hooked it on to GFP and we made this transgenic mouse. And why did we do that? That's because we could now label, as you can see from this entire mouse, you can see that only the lens is labeled right there, right? And we could now start dissecting that lens from the very early age. This is 0.5 millimeters, and this is done by really not any laser capture microdissection. It's really done by sophisticated, just dissection from your hands, okay? So microdissection is what we'll call it. So we first, convinced ourselves that we could really dissect this lens tissue from a very small amount in this very early stages of a mouse embryo. But this is a mouse embryo, one may ask, and how can this really help us with human gene discovery, right? And 22,000 of our protein coding genes between the humans and the mice are similar, okay, or we have orthologs for the mouse genes, okay? So that means that for the protein coding sort of genes, we are very similar to the mouse. How these genes are wired with each other is where we differ, basically. So the essential part is that if we could start putting a tool together for the mouse, and we started predicting these genes in the mouse, and we started testing it in the mouse, we should be able to try to use those rich pool of candidates and try to translate that information into human patients, okay? So that's the principle. So we could dissect this particular part at a very early age, the lens being formed as a vesicle. We started dissecting other stages, and these are 3 representative stages I'm talking about in the mouse. So E stands for embryonic day, 10.5, 11.5, and 12.5. This is when the lens is forming from a surface ectoderm. It's trying to make a vesicle and then sort of close itself, and that's when the fiber cells are being differentiated. Very critical time points, if you can think about it. If you don't have separation of the lens and the cornea, you're going to get a feature that is seen in humans called as Peters anomaly. Okay, there's going to be a stalk between the cornea and the lens. So all of these stages are critical for understanding human developmental defects. Okay, so of course the next part was relatively easy for a developmental biologist or a molecular biologist. You make RNA from these lenses, and then you basically send it for microarrays, or you can send it for RNA sequencing. This was done a few years ago, so we used microarrays, whole genome microarrays. That means that at one snapshot, we could just ask the question, out of the 22,000 protein coding genes, which genes are turned on and which genes are turned off in the lens, okay? And that's very important information, right? But we went one step further, because again, I would like to stress this point. When you do these kind of techniques, your problem is that it's not getting data, it's getting a lot of data. And how do you go from that lot of data into the most meaningful data? That's really the challenge. So we really used a concept from 8th grade chemistry, okay, and really believe it that's what worked the best. And the concept was really based on molar changes. Look, so 1 molar solution is 1 gram molecular weight of a substance in 1 liter of water. That's 1 molar solution, right? So we just thought that if we look at the lens, and if the lens is a small tissue and it has, say, a certain amount of PAC6 transcripts in there, and if you look at the whole entire embryonic body, maybe the amount of PAC6 transcript, PAC6, I'm just giving as an example because it's an established important gene in the eye, those transcripts are basically well distributed toward the body of the embryo. Okay, so what we decided to do was, if we look at just absolute expression levels of a gene as important as PAC6, you'll be surprised, but they are not as as basically exciting or amazing to look at. Okay, that means that you don't need a lot of PAC6 protein in the lens to do its function. Okay, a few molecules will do. So one can just look, if one does this kind of a microarray experiment, and if we ask the filter that, hey, I'm just going to look at what is highly expressed there, you might miss out on a very important candidate like PAC6. Okay, and so how do you not miss out on a candidate like that? So here is where we thought that if we don't ask the question what is highly expressed, but we ask the question what is highly enriched, okay, and that's where the molarity concept comes in. That means what is higher in molarity in this small tissue right here, which is the lens, then probably that would be telling us that the whole list of genes that we are going to be looking at is going to be completely turned around. Okay, suddenly you're not looking at genes which are just important for any eukaryotic cell or that matter mammalian cell to keep itself functional, things that are important in glycolysis, metabolic pathways and such. Now you're certainly asking the question, what is important for this particular cell type to retain its identity, to make its identity and such. Okay, that's the developmental or differentiation associated genes. So essentially, how did we solve this problem? We really thought that we should compare it with the embryo as the embryonic genomic DNA is expressing itself. And what I mean by that is that we took a series of embryonic time points in a mouse developing embryo, and we thought that the embryonic development time points when the genome is expressing different genes in different tissues, and it's trying to make all these different tissues, you're going to, if you ground it up, you make the RNA out of it, and you basically make a microarray data set out of it, you're going to get a ground level, basal level sort of control. So the molarities of all the 22,000 transcripts in these this ground level reference data set, let's just call it that right now, is going to be at a normal sort of molarity, at a sort of basal molar levels. And now you can compare any tissue of your choice. In this case, the first example, we'll talk about the lens. We can compare that tissue of our choice, and we can try to see whether with a simple t-stat sort of comparisons or a p-value on the based on fold change comparisons, we can just try to see which genes are highly enriched in the lens compared to the whole body. Okay, so enrichment is what I will focus on. So how well does it work? So again, based on the concept of a chemical concept of molarity is what I want to say. So if you take the top 500 genes based on their absolute expression, that means just ask the question, which are the most highly expressed genes in a lens at a certain time. In this case, 10, 11, and 12 means those 10, E10.5, 11.5, and 12.5 samples. And if you ask a question, well, if I throw in another sort of engine, bioinformatics engine, to analyze it and tell me what do you see in these 500 genes, what kind of genes are enriched, what the bioinformatics engine will tell you, which is online for anybody to use, something like David analysis, for example, it will tell you that, oh, these are genes which are important for really glycolysis, or they're important for translation or such. So mostly genes which are housekeeping, okay, that are important in any other cell of our body probably, okay. But then you do pick up a few of these genes which the engine will say, hey, maybe this is important in the lens. The crystallins will come up because they're very highly expressed, right? But you can already see that if you had the top 500 list of genes, and most of them are housekeeping, you're missing out on the most important genes that are relevant to human cataracts or mouse cataracts or lens development, right? But now, if you do this, what we call as in silico subtraction, which means that comparative analysis between the tissue of your interest, that is the lens, versus this whole embryonic reference data set that I just mentioned, like the grounded up embryos, then this changes dramatically. And with subtraction, suddenly all of this haystack is what I would like to call it, the genetic haystack, is thrown out, and suddenly you are left with the needles. So we are not searching for the needles anymore. We're just throwing away the whole haystack. And now we are looking at what is left out of that. And suddenly you can see that these genes are very enriched, and now suddenly these genes with lens function can be well recognized by search engines and such. Okay. So this is cool, but how does it work with what we already know with the lens, right? So if you overlay some of these candidates with what we had already seen from this literature-based, evidence-based sort of gene circuitry diagram, you can see that most of these nodes, which means the genes which are connected with each other by edges, which are the interactions between the nodes, okay, these genes could have been found by this system that we now call as EyeSight, or Integrated Systems Tool for iGene Discovery. Okay, so that's cool. We could retrospectively say that this would have worked well, but how can we use this for a disease gene discovery? That's what our goal is to begin with, like, for example, cataracts. So to do that, we did another retrospective analysis. We looked at that time, all the cases that had been basically described for non-syndromic congenital cataracts in humans. So each of these gene out here is assigned to a family or a set of family of human patients, unfortunately, who had inherited cataracts, and a mutation in that particular gene led to congenital or pediatric cataracts in their families. Okay, so what we did was looked at the original interval size, which is shown in the third column on these chromosomes, each of these chromosomes, okay, that the gene is found on, and you can see they range from 46 megabases to say around half a megabase. But what you would appreciate on the fourth column is the number of genes, right? Number of genes in each particular interval. So even if you do the best genetics that there is right now, short of exome sequencing, and use positional cloning to see that, okay, this is where I would say the region can be mapped between all the cases which are inheriting the phenotype, that is the cataract, you would come up to half an MB to 46 MBs, and you will still have the challenge of going through hundreds of genes or tens of genes before you find out what is the important gene that was probably which had the mutation which was causal. Okay. Now, this is, of course, expedited by exome sequencing, but we'll get to that later. Okay, but so we asked the question: if we could use eyesight, okay, our tool, and we could just rank this now based on eyesight, these genes in these intervals, could these ranks be indicative of how eyesight could prioritize these genes? And you'll see, except for 3 cases where there's a rank of 34, 21, and 7, all the rest of the cases have got a rank of 1 and 2. And what that means is eyesight could completely predict in 90% of the cases to the accuracy of the top 1 or 2 genes from average of 100 genes or so in these cases. Okay, so what it means is that if you had 100 genes in a narrowed down interval, and if you pass it through this sort of filtering system, you would end up with just the top 2 or 3 genes. You'll end up with a list of genes based on priority, but generally the top 1 or 2 genes would probably pan out to be the right causal gene which would harbor the mutation for that particular phenotype. But this is great. This is, but still retrospective, right? This is looking at what we did in the past and sort of try to exploit that this particular tool would work better in the future. But what about the future, right? What about new cases, right? That's when really I can drive this home. But before I basically talk about that, I'll just say that this is now hosted as a very user-friendly first version, and we are trying to develop a second user-friendly version as well very soon. But this first user-friendly version really has a color codation heat map, as you can see on the right. And what it essentially means that you can walk on any particular chromosome, and you can basically look at any particular region that of interest of a mapped region in a certain patient or a family of patients, and you can just ask the question down below right here, which of these genes have the highest heat map, the highest sort of brightly colored heat map right there. Okay, as you can see here, this is a bead filament protein right here. This is found to have mutations in humans, and that gives rise to congenital cataracts. And you can see that's the only gene which is colored bright red, as you can see, between the entire 7 megabases interval. Okay, so this is just to show that this is how one can use this particular system, essentially just by really walking on the chromosome online, okay, for a few hours or such, okay. And so now let's get into how we can use this sort of a technology or a method or a tool or an approach to come up with new cataract and eye disease genes. Okay, so the first example, the first case I'm going to talk about is of a family which had inherited cataracts as congenital cataracts, and also an isolated case of a de novo change in a boy in Iowa who had developed cataracts as early onset cataracts at a young age. And we found the gene to be TDRD7, the causal gene to be TDRD7, and that caused congenital cataracts. So how did we first find it? So when we looked at the first case, we knew that the breakpoint was narrowed down to around 10 MB interval, to make a very long story short. And we really just combed that entire region with eyesight, just as I told you right now. And eyesight predicted that this gene called as TDRD7, okay, would be the most important candidate. You can see it's the most brightly colored candidate out there. That means it's highly enriched in the lens compared to the rest of the body, right? So at that time, I really was, I didn't know where to go, because I said, okay, eyesight is telling us it's TDRD7, so let's go after it. But as a biologist, I would have never gone after this gene, because all that was known about this gene was that it was important for germ cell development throughout metazoa, throughout the animal kingdom that we know of. So from worms to humans, TDRD7 or Tudor domain family of proteins were important for making the male or the female germ cells. Okay, so why would anybody look for it in the human eye, right, for that matter? But we did look at it, and again, making a very long story short, this is the actual case, this eye of the patient who had the pediatric cataracts. We could find that haploinsufficiency in this one case, or mutation in the whole family of in recessive inherited cataracts could give rise to congenital or pediatric cataracts in humans. Okay, we screened for a mouse mutant at Jackson Labs and found one mutant which was missense mutant. That means it's a null mutant for TDRD7 that got cataracts. And then we collaborated with another investigator in Japan who had made germline knockout, targeted knockout mouse mutants for TDRD7, and that got cataracts too. And these cataracts in the mice were 100% penetrant, almost like a spot on, switch on at around 3 weeks of age after birth. But we are now seeing that many things are going wrong even as the lens is very younger before you even see the phenotype in these mice, and I'll get to that soon. And of course, we knocked it down in chickens, and you see that TDRD7, when you lower the amount of TDRD7 in chicken embryos, you get a cataract as well. So this is fundamental to vertebrate lens development. And of course, the reason we picked up this gene, if you do an in situ on this, you can see that this is the entire section of this mouse on the left right there, and the middle one shows that the gene, which is stained in blue because of the probe that we use, is stained in a certain colorimetric way. But you can see that it's expressed specifically in the lens, and within the lens is specifically expressed in the posterior part of the lens, the fiber cells, which are going to produce a lot of crystallins and are important for transparency. And that expression is retained in chickens as well, which is shown in the right most figure right there. Another cool really result that we saw here was that Ramon y Cajal, an amazing cell biologist from Spain, over 100 years ago had described these structures called as chromatid bodies in differentiating sperm, okay, in animals. But really, we are just about grasping as to what they're doing for spermatogenesis. So as I told you before, TDRD or the Tudor domain proteins were linked to germline development. So when we stained the mouse testes, and this is a snapshot of that on the left, you can see that it beautifully stains these original bodies that Ramon y Cajal had described, these chromatid bodies, okay, all these dots that you see on the left, okay. What was surprising was that when you stain the lens, you can see similar kind of dots in the lens fiber cells as well, as can be seen on the right, okay. So what would this predict is that if you knock it out, of course, in mouse, you should probably get two phenotypes in this mouse, okay. And the one is on the left, that is the spermatogenesis defect, which can be seen that these homozygous null mice have got no mature sperm. And of course, I've just shown you the lens, but they get 100% penetrant lens defect. This is a severe case where the lens, ball of lens is, in the center of the lens has all exploded the lens and come out, okay? So it's a very severe defect, right? What is Tudor protein, this TDRD7? It's got these very interesting 3 domains which can bind RNA and 3 other domains which can bind to protein, okay, and methylated arginines within proteins, okay. So this is a very interesting protein. And why these proteins are important? Because they control gene expression not on the transcriptional level, but they can control gene expression after the transcript. That is the first, when DNA to RNA, the information is transcribed into an RNA. When that RNA is supposed to be modified, or it's supposed to, the cell has to take decisions as to when to make protein from this RNA, or to degrade the RNA, or localize it at a certain place. This is where you need these RNA binding kind of proteins to take those decisions, okay? And this is a very understudied area in research. Just as a quick comparison, we have 1,500 transcription factors in our genome. A lot of them are very well described now and linked to human diseases. Not a surprise, right? Because transcription factors are proteins which can turn on gene expression or turn off gene expression. Very fundamental, right, to read the genome. But now I can tell you that we have around 1,500 RNA binding proteins. So like the transcription factors, which are generally DNA binding proteins, we have these 1,500 RNA binding proteins, but less than 50 of these proteins from 1,500 are linked to developmental defects, okay? So that means that this is an untapped, really, territory, and a lot of interest is now getting into the grants that you mentioned right now, post-transcriptional regulation. That means what happens to the regulation of genes when the gene is transcribed, okay? So that's post-transcriptional regulation. So very long story short, just to tell you why these mice get cataracts and probably how this can be translated to the human, right? Why the human patients got a cataract. We studied the mouse knockout models in detail. And as I told you before, these cataracts appear, seem to appear like a switch turning on at a certain day in early development, right? And so in early postnatal development, that is. And so we thought that there must be something globally going wrong. Okay, there has to be something that is just completely breaking down at a certain place. So one thing to look at was the cytoskeleton, right? And of course, the cytoskeleton is critical because all of our cells have got a cytoskeleton, right? F-actin, which is filamentous actin, is important in all our cells, right? Because the cells have to maintain a certain shape, be it a neuron or be it a lens fiber cell. So the principles we'll learn in cytoskeleton by studying TDRD7 will be probably useful in other places as well. Okay, so the first thing you can see, and doesn't take any expert of lens biology to note that, is that on the left you can see these beautiful string lines, beautiful lines of actin, which are the fiber cells of the lens in this normal control. And on the right you can see that it's completely messed up to use a really normal common term. And really it's aggregation of actin that you can see there. So that somehow the actin is not maintained in these cells, okay, at this stage. So again, a very long story short, this is done in collaboration with a colleague, Velia Fowler in Scripps Institute. But what we have seen is that not only this actin defect is specific to the TDRD7 null mouse lens, but it's specific within the lens. And so you're seeing here the epithelial cells on the left. And as they're differentiating into the fiber cells, so the rightmost fiber cell is the most differentiated fiber cell, or the late differentiating fiber cell. It's the oldest fiber cell that you can find. And on the left out there is the epithelium, which is just starting to differentiate into early fiber cells. And you can see precisely at this place right here, which I have highlighted, that's when actin gets breakdown. You can appreciate that in the early part right here, okay, you'll see this part, the actin is deposited well, but almost like a switch, within a switch in some sense. Okay, this is getting broken down where these cells are undergoing nuclear degradation. So another fundamental principle, when cells are undergoing organelle and nuclear degradation, that's when it needs some sort of an extra effort to keep its its cytoskeleton intact. Okay, very fundamental. Look at it. And we are going from a human patient gene discovery to a very fundamental cellular phenomenon in probably all our bodies, right? And so what controls it, right? What does TDRD7 control that actually controls F-actin? So to do that, we collaborated with a bunch of people, and we basically Robert Mason from Nemours actually did this experiment for us. And what we did was we grounded up lenses again from these mutant TDRD7 mutants on controls, and we made a mass spec and 2D gel analysis of that. And the spot that was most differentially expressed in these mass spec experiments, in this 2D gel experiment, we could identify it by mass spectrometry to be this heat shock protein called as HSPB1. Again, a long story short, we could find that using these experiments, we could show that TDRD7 can specifically bind to this HSPB1 transcript, or the mRNA for this HSPB1. And we found that when you look at HSPB1 staining in these mice, you can see that where these nuclei are going to get degraded, that is in the inner part of the cells, the HSPB1 expression is lowered or reduced. Okay, so it's not completely gone, but it's very dramatically reduced. Okay, and so the very straightforward model right now that we are thinking about is that when you are stressing the cell, that means the cell itself is undergoing nuclear degradation in the middle part of the lens. Okay, you need TDRD7 to somehow maintain this HSPB1 transcript, which is this pathway right here. And HSPB1 being a very old, ancient heat shock protein is important for keeping that F-acting cytoskeleton together. Okay, and if you mess that up, you end up, unfortunately, aggregating the F-actin and eventually getting the cataract. So from mouse to human gene discovery to back to mouse, we have found a new cataract gene and found a fundamental new insight into the molecular biology of cells or cellular biology. Okay, so of course the bigger picture was that with this one case told us that post-transcriptional regulation, I keep coming back to that, right, was important in the lens, something that we really have not studied well, not just in lens, but even in other tissues. So we should start trying to look into genes specifically from eyesight, which are fitting the bill with post-transcriptional regulation and try to see if we can characterize these genes in the lens and try to see if that knowledge can be translated to other tissues as well. So the second case that I will talk about, which came from eyesight, is this molecule called Celf1 or CUGBP1. Again, this is how eyesight predicted it. You can see that it's in the top 1 to 2 percentile of enriched genes within the lens. And it's got these 3 RRM domains, which are again well-characterized domains for binding RNA. Okay, and you can see that expression of this Celf1 gene is specific in the lens in fish, frog, or in the mouse, as you can see here. And again, they form these granule-like patterns in the lens as well, and they are also enriched in these fiber cells. Okay, so when you again, long story short is if you knock it out in mouse or if you knock it down in fish and frogs, you get profound eye defects. In some cases, in the frogs, you can get a complete loss of eye, but in all cases, you get profound lens defects. And you can see this cataract. Again, it doesn't take an expert to see that this lens is completely opaque and even, in fact, exploding. Okay, so now we understand the fundamental basis of cellular and the molecular basis of this defect as well. In TDRD7 had this actin defects. In this particular case, you can see on the left, the clear lens of a mouse which has got no cataract, which means it's a normal control, is beautifully cleared in the center. That's how it can see, right? Because there's no nuclei in the center. But you can see that on panel B dash, which is a zoomed in image of the central part of the Celf1 mutant mouse lens, you can see all these nuclei there. And this nuclei are retained in the TDRD, in the Celf1 knockdown fish experiments as well. So what it's telling us is that these lenses, which don't have Celf1, cannot dissolve their nuclei in the central part of the lens. Okay, what's the molecular mechanism of that, right? Nishimoto in 2003 had published a paper in Nature saying that they had found this unique DNase2 beta. So this is an enzyme which can degrade DNA, but they had found it uniquely expressed and enriched in the lens fiber cells. And when they knocked it out in mouse, that means they deleted that gene from the mouse, they found that these mice lenses had retained their nuclei, similar to what we had seen in our Celf1 mutants. So when we looked at DNase2 beta, we found it was 6-fold down regulated in our Celf1 mutant lenses. And when we IP'd, that means when we used a Celf1 antibody and we asked whether this DNase2 beta transcript is enriched by this Celf1 binding, which reflects Celf1 binding to this particular transcript, that turned out to be the case as well. So the model here is that Celf1 can probably bind to the DNase2 beta transcript and hold it enough, stabilize it so that it can be translated into enough amounts of DNase2 beta that is necessary for nuclear degradation. Okay, so another fundamental insight that we found from this particular mouse model was this P27 gene that I talked about, right? I told you about this P27, which is an important gene for cell cycle kinase inhibitor. This is fundamental to cancer biology, I think. Okay, so you can see that the pattern of expression during development in the cells on the left-hand side in the control, it's turned on and then it's completely shut down. Okay, so how does it shut down? Nobody really understands that mechanism. But in Celf1, we now found that this gene is not shut down. As you can see in the central part of the lens, you can see a lot of this expression of P27. So again, a very long story short, we now know that Celf1 can bind to the P27 5' UTR, that is the untranslated region, the region before the coding part of the transcript takes place, and it can hold and make sure that this transcript is not translated into protein in the central part of the fiber cells. It's a very fundamental new insight into how P27 can be controlled in developing tissues, okay, from this lens development. So far, we have not found a human mutation in the CUGBP1 or a Celf1 gene that can be linked to cataracts in humans. The reason for that could be two. One is because these animals also have cardiac defects, and if the humans have cardiac defects, then unfortunately it could be a much earlier sort of onset lethal probably mutation, or that could be one case. And the other thing is that there's an indirect evidence that Celf1 might be important, and that's because if you have an expansion of targets of Celf1 in muscular dystrophy patients, you can misregulate Celf1 in an indirect way, and that is known to cause pediatric cataracts. So there is an indirect sort of misregulation of Celf1 linked to a human, but a family of a case of cataracts in humans. But so far we have not found a mutation, so that's an area of collaboration that I want to expand and try to see if human geneticists who may have narrowed down a case of human cataracts on this particular chromosome can look for Celf1 genes, okay, or sequence Celf1, that is. The third case I'll be short on this one because I have I've got 10 minutes to go, I guess. Okay, a lot of things to cover, but we'll see. But anyway, this causes Peters anomaly and lens defects in mice. So Peters anomaly, as I said before, is when the cornea and the lens do not separate out well, and we think that we know that when we knock out caprin 2 in these mouse models, we get this Peters anomaly. And you can see on the right-hand side there is a stalk which is remaining between the lens and the cornea, and we have identified another RNA binding protein, caprin 2, which is a new protein that is again a candidate for Peters anomaly in humans. Okay, and the fourth case for this particular part of the discussion is RBM24, another completely new gene, which we know now when we knock it out in mice causes microphthalmia and anophthalmia. Okay, so you can see this mouse right here. There's no eye. Okay, and all the mice that we have tested so far have got small eyes. Okay, if they have an eye, so it's an incomplete penetrance. Okay, as a phenotype. But the most important thing here is that we found that RBM24 null mouse mutants have no expression of SOX2 in the tissues that we have seen so far. Okay, so that means that SOX2, which is such a fundamental gene for pluripotency, okay, you may remember from Yamanaka's work, who is a 2012 Nobel laureate, that if you use these 4 genes, a cocktail of 4 genes, SOX, MYC, a KLF, and NANOG, you can make any cell, my cell, from a skin cell into a pluripotent cell, right? A pluripotent stem-like cell. That means you can reverse its developmental program to where it could be pluripotent. So SOX2 is that important gene, okay? And we have found that RBM24, a new RNA binding protein, can actually control SOX2. This is important not just for the anophthalmia, microphthalmia, but also fundamental understanding of how SOX2, a very fundamental gene, is controlled. Okay, so I'll skip that, but the summary of this is that we have, of course, found several genes which are important in initial stages of lens or eye development, and also know about what they do in very specific places within the lens as well. And this has also prompted us to look for these structures called as RNA granules in the lens. This is soon to appear on a cover page of a journal where really people have not seen these structures in endogenous development. And you can see that they are in the lens and the retina. And now we are looking into these stress-like granules which might be forming in our epithelium if we are irradiating them with UV light. Okay, so this is really given our attention to other clinical aspects from this basic fundamental biology of gene discovery. I will skip that. This is really just to tell people that, hey, transcription factors are important for turning on genes, but post-transcriptional regulators like RNA binding proteins, like some of the ones that I described, are important for other decisions in the cell after the transcript is made, okay, to when to translate it and when to keep it stabilized and such. But one more summary slide is that eyesight in the last 5 years has made a huge impact in our field. It has characterized or helped other people characterize various transcriptional regulators. It has characterized several new RNA binding proteins. It has helped characterize cell membrane proteins. This PVRL3 is also a human case that I didn't describe here today because I didn't have time. But we found that a human patient with congenital cataract also has a change in expression of PVRL3. Again, eyesight helped us in this finding that. And also some very interesting new candidates like selenoproteins, CEP50, when you knock it out in mice, they get cataracts and lens defect. So eyesight has made a fundamental impact in our field in expediting gene discovery in the lens. So I'm going to really go fast on this just to give you a flavor. Can we do better than this, right, is what I'm asking. Okay, this first principle I told you was based on the chemical principle of molarity. Straightforward, very effective, worked well. But can we do better? And how we can do better is if we can put all that knowledge that we have worked as a global sort of biological sort of lens biologist, let's just narrow it down to lens biology as a field. If we can start putting together every node and every interaction between every other node, that means gene, how this gene interacts with the other gene and such, we should be able to now put new knowledge onto that and try to see what sticks, basically, really. Okay, so how do you do that? Okay, you start reading papers and you start recording data as you see here in a very systematic manner. And what this is what is the gene that was knocked out, what was the target that was tested, where was it tested, which stage was it tested, what was the way that it tested it, was it an immunostaining, what is an RNA in situ, what was it? Okay, and if you do all that and you compile really thousands of these data sets, which me and my postdoc did, we get around 2,500. This list is growing now, but it's around 2,500 from around 100 papers, where you can take this data to the bank. What I mean by that is that this is published. This has got experimental backing to it. So now what does this do? When you write an algorithm to read that, you get up this mothball of pathways. Okay, as a developmental biologist, I don't know where to start when you see something like this, right? So of course, I wanted to make this tool better for my own sort of kind of expertise or kind of people who do this sort of research. So I wanted to make it interactive. So by interactive, of course, you can play around with it. You can zoom in, zoom out. You can move around these nodes. That's very straightforward. But then you can ask the question, wait a minute, I don't see why this particular edge, that means this relationship is mentioned. If you click on that edge, you'll get some of the papers which mention that edge and why you should believe it, or you should go and read that paper to see why that edge was made. Then you can see whether, if you are interested in one particular molecule, you can look at that particular network, as can be seen here. And the bottom row out there shows you that this particular gene, if it's knocked out in any other particular mutant or gene, what effect it has. So in a snapshot, really at your fingertips, you have all this information. You can look at how genes are co-expressed with this PAC6, or genes which can bind to this particular case, PAC6, so which protein-protein interactions and such. So this tool is not yet freely available to anybody online. Of course, it will be freely available eventually after we publish it. But as you can see, the beta version of this is already out there, done in collaboration with Joshua Ho, who I published the first version of EyeSight with. And of course, this lens GRN is comprehensive, okay, because we can now ask the question, well, what happens in primary fiber cell differentiation at a certain stage? Remember, I told you that the stages were also mentioned in this informative sentence. So you can see how the network really flows and really matures as lens development occurs. Okay, so now, of course, this is all cool to look at, right? But is this predictive? It's going to tell us some new information, right? I promised you that this will actually tell us some new stuff. So how is it predictive? So to do that, I'll just highlight one case in the last couple of minutes, and then probably we'll have to stop, and before I get into the cis-face part, or maybe I'll just mention it in a minute or so. But here is a new gene that we found out, MAFG and MAFK. We found out that if you knock these genes out in the mouse, you get early onset age-related cataracts. Okay, so that they'll get a cataract at around 4 months of age, or between 4 to 6 months of age, 25-year-old in human terms. And when we looked at the microarrays of these mutants in these lenses, we could build together a network like this. But what it pointed out for the first time was these sterol and lipid biosynthesis genes were specifically regulated by these transcription factors. Okay, and we published this in April of last year, and then completely independently, there was a pharmacological approach that a person in UCSF had taken, and again, a lens genetics approach that a person in UCLA had taken, and they both converged on sterols, okay, as important to reversing aggregation in the lenses. And these are the two papers that I'm talking about, which have been published in November or July of last year, so not even a year ago, including our own paper, probably just a year. In the last year or so, we have found out that there are genes which regulate sterols, and these sterols are important for preventing proteins to aggregate in the lens. That means preventing cataracts, okay? And that means that in these initial reports, they have tried to even put drops of sterols in animal models and shown that it can prevent cataracts. So this is a huge potential. There is, of course, a drug like that exists for dogs, Kinostat, I think, but it's specifically for diabetic cataracts in dogs. But this one is going to be important probably eventually for humans, okay? So again, coming from EyeSight, basic genetics, mouse model, maybe human genetics, all that approach coming to therapy, right? That's how the journey is. I have to skip this part. I just don't have the time. But essentially, we have already extended this approach to the retina. It works the same, okay? This is just to say we can find all the genes that people had found as very fundamental genes for cone rod dystrophy or retinitis pigmentosa or the Bartter-Biedl syndrome. Many of these candidates can be found by the same approach, and we are trying to now put this together. This is also unpublished, but this will be an important tool with all the similar sort of networks and such. And now let's get to, in the last minute or so, to cis-face, which is for really systems tool for craniofacial expression-based gene discovery. So NIDCR was interested in this approach because they had made this database called as FaceSpace, but they found that it was not as used by the craniofacial community. So when they saw that we had made EyeSight and it was so useful for the eye research community, they basically gave us a grant to build similar kind of a tool for craniofacial research. And I'll just tell you very shortly, it works the same way. Again, the subtraction works. You can find the same genes. And here is a table which shows that we could find all the previous genes. But I'll leave you with one last collaborative effort that we have done with Dr. Aziz Boutali from the University of Iowa. Dr. Boutali had identified this extended family which had these several cases of clefting of the facial clefting, orofacial clefting, and really what he had done was done exome-seq analysis. Okay, so one can say, hey, I'll just exome-seq it and I'll just find a mutation, but it's not that straightforward. That's what I want to give you by this particular data. So Dr. Boutali found around 30 variants in there, and he didn't know where to prioritize this, and we used CISFACE to prioritize, and you can just see that the most brightly red color right here is the candidate which turned out to be the right candidate, called RGAP29. Okay, and so CISFACE is now being hosted, again unpublished, but being hosted. Already the craniofacial community, there are several collaborators who want to collaborate with us on this particular aspect of it to really try to find out new genes which are linked to craniofacial clefts. Okay, so in summary, systems tools like eyesight or CISFACE can be used for new gene discovery. Fundamental new insights can be gained. In addition to discovering these genes in the human patients, they can really tell us more about how our body functions in our cells. And as I've shown you with several examples, P27 and actin and such, they can tell us some really fundamental information of regulatory information that may be applied to other tissues and such. And again, take-home animal models and this can be used for prioritization from all sorts of analysis, from exome-seq to GWAS and such. And this that I think I've shown to you that it can be extended to other tissues. So I would like to thank, of course, the funding that made it possible, and of course the people who worked hard to make this possible. That's my team in the Lachke laboratory. They've been acknowledged in each of the slides where they contributed. So in the interest of time, I won't go through the whole thing again, but a variety of collaborators and funding and lab members who really make this possible. So thank you very much for your time, and I'll take any questions.
When you work on the network, the network of the eyesight that you showed us, if you discover another gene, how do you integrate it back into your network?
So if I basically discover a new gene like say TDRD7 or such, the network is kept growing. So one has to every year or so update it. So that's a strategy right now. I mean, this is current as is right now. It's very current right now. But you're right. Every year, or say at the end of every year, probably one has to go back and curate it or update it. And that's what happens with these kind of databases right now.
So is it? Do you do that, or is there?
So nobody has. I mean, we have built this right now. So probably the responsibility is going to be us doing it.
And
yeah.
Will you do it each time you find another gene, or will you say, well?
No, I think we'll do it every set time. I think. We'll have to say that. This can't be done on a daily basis because we have other things to do. But I think every December or so, one can reflect on what was done that year and make an update out of that or something like that. Unless we get more grants to keep maintaining this, then we can do it. We can have a dedicated person doing it in a more real-time basis.
I had also another question.
Yeah.
The mutation, the TDR7, is it
yes.
TDR7 mutation that you found related to cataract, and you said that this gene was also involved into spermatogenesis.
Yes.
So in the children who have congenital cataract, do they have also defects?
We don't know yet. The patient was 2-year-old when we got that, and the patient is not yet reached puberty, I think. But that's the ophthalmologist who worked closely with this, who is on our paper, published paper. They said that it will influence future sort of medical care. But we don't know yet. The short answer to that is the other kids that was from the Kuwait, the other family. I didn't really show you the entire genetics of that, but there are 4 affecteds in that family, and those are small kids too right now. So we'll just have to wait to see, sadly. It's an important point, of course. And it also shows that this can be translational in the future in future aspects of the patient care.
Great. Thank you so much.
All right. Thank you.